Oh, wow. Okay.
So as usual, everyone, I've dropped a couple of links in the chat. The first one is a small form so I can gauge who is turning up to the sessions and we can hopefully make improvements next year. There will be a more of a feedback form at the end of BGA as well. So you can give your overall feedback of how well we did or if you think we should make some specific improvements. There's the GitHub link, which will take you to the Git pod as well. And then there's the Discord link if you don't already have it. It's if you can use the Discord for questions, that way we can keep the answers for perpetuity in case anyone else comes across the same issue or has the same question as you in the future. Right. Okay. Should we get started, Camille?
Sure. I hope to tweet the live stream link, but it's okay. You can just ping me in a message once it's up.
There you go. In the
Oh,
chat.
is it? Perfect. Three, two, one, go.
Right. Okay. Good morning. Hang on. I need to set the recording.
Uh-huh.
There we go. Right. Good morning, everyone. Welcome to session 13 of BGA24. This session is going to be on Genomescope by Camille Yaron.
Mhm.
Yeah, cool. Um, Take it away, Camille.
Right. So, hello everyone. My name is Camille. This is the second year we're running this session, so I think it will be a lot more polished than last year. No, it won't. The point of this session today is to learn a bit about k-mesh and about how to actually use them to look at the genomic data sets. So, uh, while I'm the only teacher of this session, I would like to give a big shout out to Lucia, who prepared all the materials with me last year. So she deserves at least a partial teaching credit this year too. Right. So this session will have two unevenly spaced halves, um, but I think I would rather make it a bit more interactive. So based on actually how you will be doing, we might adjust of which one we pace through and which one we spend time on. We will go through the very basic examples of how to actually just fit some KMS spectra with some genomic models using Genomescope. I will try to get you to fully understand of how the model works by making you actually reconstruct the model yourself in base R, which you find that it's actually surprisingly doable. And then I, as always, prepared a beast theory of a really weird chemospectra we can just together look at. It's I would say by far the most popular part of every workshop. Um, I really do appreciate everybody having their camera on, so keep it up. It's and keep nodding and reacting and or interacting, all appreciated. So why are we here today? What is the deal? Well, sequencing, as much as we would like it to just read the genome as it is, it's still sort of generating us sort of this pile of reads, so, which it's a bit like what we want is that one stem of a grass, and what do we get is the pile of hay, and we now are given the task to reconstruct it, which is called genome assembly, and that exists. There are algorithms for that. You all know that, but do you actually know how any of them truly works? Like how they make every individual decision? It's actually a very complex problem. It really depends on what you throw there as an input, and this will give you the output, and it depends on lots of biological properties of repetitiveness, ploidy, heterozygosity, and so it's too black boxy for genomicists to actually truly just do genome assembly. So that's why we have this completely alternative way of how to actually look at the data, which we call genome profiling. This does not attempt in any way to reconstruct the genomic sequence, but it's aiming to characterize some of its genomic properties. So in the analogy, it would be something like estimating how many grasses were actually cut into that one pile, or how many seeds there were on average on each plant. And but the question is how to actually do how can we look at this bag of reeds, this pile of hay, and before we actually get into the cameras and how to actually do that, I would just like you to appreciate of why it's actually necessary. What is the alternative? Imagine that you would like to compare only two sequencing reads from a genome. Okay, how would you actually go about it? What would you do? Would you try every single possible overlap, and then what would you do? Would you calculate if they perfectly match or not, or would you if they're perfect reads of the perfect template, I guess that would work. But what if they have sequencing errors, right? It becomes immediately non-trivial and computationally actually fairly complex task, right? And this is just simply being able to tell whether two reads came from the same genomic region. So, full disclaimer, this is not a problem that doesn't have a good solution or an obvious one or one good solution. But the funny part is that every single solution that exists that actually does internally use KMS in one form or the other. So although you might feel like you are actually aligning reads, you're not using KMS. Actually, what's happening inside quite often and boils down to KMS. So what the KMS are? The concept is actually fairly simple. It's KMS are by definition the substrings of length K contained with a sequence or biological sequence, which practically means that you just take a read and you chop it to evenly sized bits on a sliding window of a certain size, which in this example is 7. This choice of 7 is arbitrary. It can be 1, and then you would have just individual nucleotides. It could be 4, and you would have just 250. Ooh, 2 powered by 8. Well, it's it's a few hundred possible kmers. Or if you get in the case that are actually a bit larger, like 21, you get to these astronomical numbers of how many theoretical kmers actually are. And those are actually the kmers that are starting to capture a bit of a complexity of genomes. Basically, what you actually want is to have a certain fraction of a genome be represented by unique kmers. So why kmers work and reads do not? If we even if we manage to do those comparisons, we already agreed that it's somewhat problematic just to simply use perfect match. Perfect match is what we want to do because it's the very speedy thing how to compare sequences or anything really. Perfect match is what we know how to do computationally. But if you would look at the fraction of reads that contain an error, it actually would be fairly high. But if you take a read and chop it down to all the possible subsequences, only those subsequences that are at least the k distance from that error will inherit the error, while everything else will actually truly represent the genomic template. So the fraction of errors, or fraction of reads that contain an error is much, much higher than the fraction of k-mers that contain an error. Does that make sense? Good. So these true genomic k-mers, this is what we are after. This is the essence of why this all spiel works. I'm getting lots of Discord notifications, but I will not be paying attention to them, and I will just rely on Damon to. Okay, I because I cannot even tell. I am on too many channels, so I'm not even sure which one is ringing. Well, right. So we have a bag of reads. We know what to do with each one of them. We just take all the possible subsequences of the certain K. So, and then we just simply count how many times we see each one of them. So, in this example, this keyboard was seen 28th time in the whole bag of reads and etc. Well, and then if you plot a histogram of these coverages, then the magic happens. So let's just very slowly unpack that plotting that histogram. So on x-axis, we will have a coverage. On the y-axis, we will have the number of gamers that are at that coverage, right? So we have a genome that is perfect, say diploid, that's fine. It has perfect sequencing, and all the regions within the genome are perfectly unique. So how would the K spectra look like? Does anybody have a guess? It would look like a single peak. And this peak would be the coverage depth variation that just simply comes out of the sampling process. When we're sequencing, we'll randomly pulling reads from different regions of the genome, right? If you do it sufficient number of times, you will have a distribution of coverages per region, right? Makes sense. So, but what if we add there the sequencing errors? Where they will project? Well, it's actually fairly easy because the errors, although they can occur in the same genomic location, they are hardly ever the same nucleotide, and even if they are, they're hardly ever the same type of error. So they will be all much more rare than the genomic gamers. So they will just have this error tail on the left. Of course, genomes are not perfectly unique everywhere. Sometimes it just happens that you have a duplication. What would a duplication do? Well, it would create this higher frequency gamers. Quite specifically, if it's a duplication, it would be at twice as frequent, twice as high frequency. Well, sorry, twice as high coverage. And perhaps while we are at this, now we are unpacking the whole concept of K-mass spectra, we can slightly talk about how does it relate to the trace of K. So I said that we pick those Ks that are big enough to capture the genomic complexity, but there's a bit more to it. So basically, every time you choose a K, every repetition that is longer than that K will be collapsed on that K, right? So if you use K31, all the transposons that have 45 base repetition will just uh, be repetitive. If you will have a K that is 51, then every single of those repetitive regions will also have the flanking bits around. Not all of them will be unique because there will be just few nucleotides, but it will give you something, right? So basically, more higher K you choose, more regions within the genome will represent a unique sequence. So basically, higher the peak will be. However, higher K you will choose, less Kmers there will be in total, which comes just simply out of the fact that if you have a read and a bunch of subreads of a certain length, the longer length you choose, fewer reads you have there. That's one aspect. And second is if you have an error, it will propagate to that many reads as that many Kmers as the K length because it just affects all the Kmers before and after, right? So basically, the number of genomic Kmers is going to decrease with increasing K. So this is a trade-off. So increasing K will increase the size, but move it to left. That's a bit I should have written myself a note for. The the choice of K regarding the it's sure okay if uh, fewer the Kmers there are in the distribution, narrower is the peak. Okay, actually, I will not even try to elaborate on it. I remember asking Lucia about it last year because I didn't get it out of slides, and then she explained it and made sense. But now I cannot reconstruct it. Okay, my apologies. We need to ask Lucia for that one. It's so the sequencing variation actually does relate to K, but it's I would say it's actually not that trivial. But that's oh, I think this is what it. Okay, okay, I got it. So if this is the very same data set, just run on lots of different Ks, just simply to demonstrate of how it works. So you see that for very low case, yeah, it's sorry, for very high case, it's a low, it has low coverage. For a very low case, like 21, it has very high coverage, okay? But you also see that for K21, the peak is very wide, while for K121, which is the leftmost peak, it's very narrow. So I think this is what Lucia was trying to say, that the sequencing variance is actually decreasing with lower sequencing coverage, which is perhaps a bit unintuitive. Right. So we were talking about how the chemospectra looks like for a diploid genome that has no heterozygous loci, and that's actually very unreal. So what it would look like if we added just one last thing? And I hope that that will be no surprise to you at this point, that it will just generate one more peak that will be in between of 0 and the 2n peak. It's 1n peak. It should be exactly the half of a coverage. So out of this kind of chemospectra, you can estimate the genome size if you just simply look at the number of reads and how many chemos there are per read. So this term on top is how many chemos there are in total. And if you divide this number by the estimated the chemo coverage, and multiply it by ploid, you will get actually the estimates of the size of a haploid genome. So okay, so now we are reaching the point that there's very little to describe theoretically on the chemospectra, and now we would like to start modeling. So how does it work? So you take your sequencing reads, you count the chemos using one of the chemo counters. So people quite often use jellyfish because it's historically probably the most used chemo counter, but we all care about the environment, and perhaps we should use more efficient algorithms that are do exist and are available. So I would, if you're not working with bacteria where it really doesn't matter, I would recommend to use KMC or FastK, which are the two very speedy algorithms of how to get the KMS counted. And you fit there in a tool that just estimates the genomic properties, which is what we call genome profiling. And here we're talking about Genomescope, but there are tons of other tools that exist. For example, Respect is a tool that is optimized for very low coverage data sets. But there is a challenge that it's assuming low heterozygosity. So basically, every single one of those tools has some limitation. While Genomescope really requires high coverage, it actually converges very well for a wide range of biological features of a genome. Respect is very restricted to biological features, but can converge on 0.5x data set, which is actually very impressive. Uh, Here, I just would like to highlight the fact that this Genomescope is not the only way how to model genomic properties of a genome, and I would like to encourage people to explore the diversity of them that are out there, because the field will not move forward if we will all do the same thing. Um, right. So Genomescope is a very popular tool. It's a oh wow, that's a very old screenshot. It has a lot more citations for the record. Um, it came in two generations. The first one was only for diploid genomes. The second iteration of Genomescope improved lots of small things like genome size estimations to be more precise, or convergence of the model in general. And mostly it actually developed a complicated combinatoric model for ploid level for up to 6. So this is actually pretty neat that it actually works for all the plants genomes where you actually really need the most to actually have a nice model. While if you have like a tiny diploid genome, you probably will just be okay assembling it, right? It's a more complicated genome you're dealing with, more it's important that you actually understand the data first. And those are actually very widely used tools. So it's basically a routine for most of the sequencing projects to just simply QC to generate the histogram and run Genomescope just to see how it actually looks like. Um, you can use any type of data for this kind of profiling, really, but obviously, if I have most of the profiling techniques require high coverage. It's good to have accurate reads. If you're taking old PacBio data that used to have this very high error rates, which you probably don't remember anymore, um, then it just looks bad. If you well, it's really just function of the fraction of errors. If the error rate is over 1%, then it's becoming a big mess. And it's actually quite funny that although the PCR is surprisingly still improving. I remember when I started with profiling in 2014, 15, I could have almost tell you how many PCR cycles there were in the library prep. I could tell it by the width of the distribution. that I could just say that for this coverage, I know that it's usually not that wide. And but it's over time, it's actually better and better. The PCR is harder and harder to spot. So uh, I guess the general rule of thumb is always try to minimize, like less PCR there was involved in sequencing, nicer the chemostructure will look like. It's 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 actually much less of a truth than it used to be. So Genomescope is running also. You don't actually need to learn any command line. You can just do it in an online environment. There is a web service, and you can just simply go to genomescope.org. This URL should be updated again. I will do it later on. You can just drag and drop the KMA histogram and just put there some parameters, and it will just generate you those plots. But you can also use command line, and it has a lot more options, and it has a lot more versatility. So I would actually recommend you to do that if you can, if you're not somewhat resistant to the idea. Um, when we talk about the histogram, it's actually not anything all too complicated. It's just a very plain text file that for each coverage gives you how many distinct schema there are at the coverage. It's as simple as that. So basically, this is the input, this is the output, and the model will sort of gives you like that. The genome score will just give you a report of the estimates of all the properties. So the, the heterozygosity, the genome length, the repeat length, fraction of unique sequences, etc. Right, and if you want to dig into it, you can actually see of what how exactly the model was constructed, and we will dig a tiny bit into that. So now I would like to get your input. So just looking at this schema structure, do you think we did well on sequencing this genome? Do we have enough coverage to assemble it? Why? Wilco, you shake the head. I know that you know the answer.
Well, since there it fits the model and there is two clear peaks, it shows that it has enough information to be able to get good context for the assembly.
Yeah, I think that's exactly right. So the whole point is that just looking at this stupid plot, you can kind of tell that errors and the template sequences within this dataset are distinguishable. They form unique peaks. They look like, it could work. Why not? I guess you see where I'm going with this. So the different gamma spectra. So this, although I for some reason pick a picture of Holly. This is a genome of mistletoe. I always realize it when I present it, never when I'm making this one. This is just to show that actually this peakness is a property that's completely unrelated to the genome size. So this is a 93 gigabases large genome, the common mistletoe. Like think about the sheer volume of data. That was like what was it? I think the raw reads had more than Tera. Uh, um, you have a lot of things in between. For instance, here in the example of the crayfish, you see that the peaks exist, but they're blended. So this assembly always will be messy. And it almost doesn't matter what technology was used. Like you know that distinguishing between what is an error and what is a genomic chemo will be just complicated. If you see something like this, so this is a leak. Leak genomes do not have 200 megs. So this is most likely that we just don't have the coverage. It's the model did not converge on anything meaningful. The real coverage will be probably something like, I don't know, 0.01. And if you would actually uh, put that in an, you remember that equation for estimating the genome size. Basically, what you can do is you can just take the coverage that was estimated, multiplied by that, and then divide it by the real coverage, and get what would be the genome size, right? And you will actually see that this is why if you cannot estimate the coverage right, you cannot have the genome size right. So when you have a completely nonsensical genome size estimate and it looks like a hockey stick, you know that you probably don't have coverage. Not always. So here I just put the tardigrade. where actually this one black line that you see is actually the real genome of the tardigrade, and it just happens that it's just covered with a thick layer of contamination that just blends the peaks together. So this one actually assembled. And the big difference between them is that actually it estimated the genome right, the genome size right. But again, don't this will require lots of understanding of your data and digging around. So this is not really that Genomescope can save you or genome profiling can save you, but it just tells you something with your data. It tells you that at least you need to be cautious, and that's good. So, are you ready to fit some models? Are you ready to fit some models? So I would like to get a bit of a sense of where you are with this. Like, how many of you have actually tried to run that software before? Would you lift your hand? So one, two, two. Good. Oh, three. Perfect. Um, for the three of you, was it okay? Like, did you? So why are you here? What are your? Oh, Melanie, tell us.
Yeah, so I fitted it to it was an undergraduate practical and they sequenced bacteria
Oh,
and
cool.
the sequencing was actually it was very shared, so the camera plot was I think like a bit weird because the sequences were just like way shorter than expected. Um
Excellent. So are you
yeah.
here to understand what has gone wrong?
Well, I guess like that was one of the applications that I've seen, but I also want to apply it to kind of like bigger genomes and more complicated genomes with like heterozygosity and different
Perfect.
ploidy and stuff like that.
Perfect. Okay. So in that case, I will probably just let you try it because it's not that complicated. So I will ask you to uh, open your Gitpod. I didn't. Did I make a slide out for that? No, I did not. So I will just show it. So if you go to the BGA Academy GitHub, there is the tutorial in the README file. It's all in one file, so you don't have to search for anything. There's lots of plots, lots of commands, and if you go through the tutorial top to bottom, you should be able to do practically everything. I try to make it a tiny bit interactive, so I'm not putting every single command, so you can just simply copy paste. You need to think a tiny bit and actually read it. But you should be able to do things. And the environment we will do it in is, you don't see my screen, do you? You do see
Oh,
my
we
screen.
could, yeah. Oh, I could.
It's hard
Okay,
because it's trying to.
now you should see my screen. So if you click on this, if you just go to the BGA Academy Genomescope, I can post the link in the Raymond can post the link in the chat. And click on the open in Gitpod. It will just start you this kind of window. If you're logged in your GitHub account, they continue, and it will just start you a new session.
Whilst we wait for that, there is a question in the Discord, if that's all right.
Yeah, sure.
So, are there any specific recommendations depending on the type of reads, short or long? And are there any Kmer values to use in particular?
Can you hear me?
Yeah, actually there are. If you have low coverage, so for short reads, changing the K actually affects the coverage more than for long reads. Why is that? I will just leave on the as a homework, or you can just read some tutorials. Like it's not that complicated, but it just takes a few minutes. So quite often, if when I had this very low coverage data sets of Illumina, and I really wanted to get the genome profiling model right, I just lowered the K to up to 17. So when I really needed to ramp up that coverage, I wanted the Kmer coverage to be as high as possible. So if you're struggling with coverage, you just go low. if you don't, and then just go for like the default 31. Actually, for PacBio, usually my default is 31. For Illumina 21, just simply because the choice of K affects the Illumina more. But you don't overthink it. It actually doesn't matter. It should not matter. Actually, it does like a tiny bit matter if you just if you were really poke into this and just do a chemospectra of all the possible Ks and do fit Genomescope models with all the possible Ks. In theory, it should cancel each other out, but you will get gently different values, and there like it still needs to be properly understood of why exactly is that. But I think these are biologically non-meaningful differences. It's a so just don't overthink it. It's a this is just okay, you know. Go 31. Don't sweat it. Right. So in the meantime, the Git pod have loaded. So the whole point with the Git pod is that it just has this YAML file that sets up the whole environment. And so yeah. Sorry. I just want to show you that if you go to the repository of oh boy, this repository of Genomescope, there is this Git pod YAML file, and it shows you what exactly happened. If you open the Git pod, all these commands just get executed, and all it does is just to have this vanilla installation of Linux, and just simply fetches Conda, installs Conda, and installs a few tools, and you see exactly how. So if you would like to install the tools on your computer, actually the easiest is just to go through this, or you can just open your own Git pod. But the reason why we have the Git pod is so we don't have to struggle with your installations. Okay, so now you have a workspace, so you should be able to look what you got in there. You can find that we have a bunch of histograms in here. See? So I would just say go nuts. Genomescope is installed there. You see? So if you run the tool and give it, oh, these are just reads. Where are the histograms? Oh, probably Genomescope. Yeah. So let's say Bombina. Oh, Bombina. Okay, 21. Blam. Just that's the thing. Are you going through the tutorial? You're not waiting for me to stop sharing, right? I see that at least the people that have cameras on are looking at a different screen. That's good. We should. Guys, it converged. So I just said that this is the input and the output should go in a directory called Bombina. So it created the directory Bombina. In the left panel, you can actually see all the files. So you can actually click on the linear plot and there you go. It fitted the genome model. Does it look okay? Did it converge right? Doesn't even know what is a Bombina. It's a fire belly toad. It's a fire belly toad. And yeah, it is right. They do have 8 gig genomes. It's a nice data set. So 70x coverage. Right. So I will give it 12 minutes of silence so you can just click through it and just play with it. Then we will regroup and you will tell me what you have found, and then we can venture forward. What do you say? Oh, good.
There's another question, if you don't mind.
Okay.
Quite possibly $1 million question. Why is the Kmer value always an odd number?
Oh, that's actually a very easy answer. It's because I actually did I remove that slide? I did remove that slide. I totally did remove that slide. Okay, unskip slide. So, we say k-mers, and but what we actually usually mean are canonical k-mers. And the canonical k-mers are the k-mers where we collapse the reverse complementary sequences on each other. Just simply because when we're sequencing, we never know which strand we're sequencing, right? So technically, reverse complementary sequences are the same thing, right? Unless you have the absolute orientation of the chromosome, but you never do on a read level. So, because we know that when we're counting k-mers, we just always flip them around, and we keep only one. And but when you look at the even number k-mers, even numbered k-mers, you see that there is always this one bastard that collapses on itself. Actually, not only one. So 80 is reverse complementary, still just 80, and this just makes a statistical mess. If you have an even number, no such k-mer exists, and this is why odd number. Sorry, that's why we just use odd. And actually, for lots of applications, it would not really matter. It's almost like a Piet thing. Like I would say we just all agree that let's just all do just odd k-mers just because for some applications it makes a huge difference. If you try to write an interpreter for the Brown graphs and you making it odd or even makes a huge difference because the even will start giving you all those bloody loops and you will have to figure out how to go through. It just happens that the odd numbers have better statistical properties because they don't collapse on each other on reverse complementer itself. Does it make sense?
There's a
Yeah,
green tick on it, so I'm assuming so.
Okay. Now I will leave you 9 minutes without any input, so you can focus in silence. Nobody talks to you. Okay. All right. Anybody found anything exciting or disturbing?
The strawberry is definitely interesting.
Mm-hmm. Mm-hmm.
Is it the strawberry is a very homozygous octoploid, isn't it? So
So somebody's just not looking at the Genomescope. Somebody's just making up stories based on what they think about strawberry.
maybe.
Let the data speak to you. Does it look like an octoploid to you?
No.
So while strawberries are diploids or tetraploids, and only the cultivar is octoploid, and it's sort of like a wild cross of multiple different strains. So the suspected genome arrangement is about A, B, B, C, 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 C. But
Right,
that's
okay.
but that's we're talking about Fragaria ex aranasa, which is like the strawberry. Lots of those other strawberries are just diploids, tetraploids.
Okay.
The one that you're looking here is diploid. I will actually. Before I will share screen, maybe I will just share the whole desktop. Can you see the strawberry? So is the left one the one you got?
It is indeed, yeah.
Anybody objects to the one on left? Okay, now we can use bits of knowledge about strawberries. What is the size of a genome of a strawberry? Oh, and everybody knows it's not 100 megs, right? The 211 is like a very common strawberry genome size knowledge. Uh, I expect that if you're actually sequencing strawberries, you know that. So I imagine that if you, as a strawberry person, fit this Genomescope profile and you say, well, 13% heterozygosity, 100 megs genome size, there's something off. It's just simply that model didn't converge well. So it's important to realize that if you're fitting multiple negative binomials to a distribution like this, there is no analytical solution. This is not an exact calculation. This is not something that can be done right. The only thing you can do is to try. You can try to converge there using a heuristic algorithm. And it just sometimes happens it converges on a different local optimum. And this is what happened here. It just actually the shifts of coverage by a factor of 2 is probably the most common type of an error for a decent chemospectra. So it's just simply just thought that this one is more likely than this one. But you can always manipulate Genomescope. So you can, for example, give it a Camer prior, which what actually mostly does is that it steers the heuristic algorithm to properly explore that space around the coverage. So it's actually it's not really bending the statistics, it's just giving it the right direction towards the genome. And it does predict you the genome size, makes sense. It's actually fairly homozygous, frequently self-thing plant, so makes sense. It has quite a few duplications in the genome, which is perhaps surprising given the small size, but this is the right model. All good? So next time you will see a strawberry and it will give you wrong genome size estimate, you will not be sweating. You will be like, huh, I know what to do now. Okay. How many of you have actually used linear regression before? Okay. How many of you used mixed models before? Okay. How many of you have used nonlinear regression before? Well, that's happening today. So this is the heuristic I was talking about. This is why I would like to demystify the whole. Hey, how did we get these numbers for you? So we will need to just do one step back and just re-evaluate of what it actually is we're fitting. Well, so we would like to fit something for the errors. We would like to fit a bunch of negative binomials for all those peaks that are in there. So if we just simplify our life for a bit, let's ignore all the multiplicity cases and just fit 4 peaks just to capture, or maybe let's just fit 2 peaks. Wait, we will. I think we will. So let's ignore the errors, because what we can just say that, ignore everything that is lower than a certain coverage, and we can manually select that. So if you're actually looking at the data, you can very clearly see that in this data set it will be less than 5x. So we can just mask it. That's no problem. And to be actually completely honest with you, Genomescope does more or less the same. It just does it a bit more in a smart way. There is no explicit term for errors in Genomescope. It's calculated as a residual to the model. And to make our lives even more easy, just let's say that we model 2 peaks. It will explain most of the data, so it's fine. So how do we model those peaks? So sampling process is Poisson. If you will be drawing balls out of a number and you expect a certain distribution of blacks or white balls. Well, anyway, sampling process is fairly well documented, but it just happens that sequencing is not a simple sampling process. It has a higher variance than you would expect if it was. And because of that, what we do is that instead of taking Poisson that has a mean and the variance linked by a single parameter, use negative binomial, which is nearly like an extension of Poisson, where you can actually model these two parameters separately. And there are different ways how you can parameterize negative binomial, but we parameterize it as a mean, which is lambda, and the variance will be lambda over what we will call an over-dispersion parameter. So if this over-dispersion parameter will go to 0, this whole term will go to infinite, and then the negative binomial is the same as Poisson. Okay, all good? It's actually, and there will be a good reason why we express it like this. You will get in a second. So we have a probability that goes by relative size of one peak, which would be alpha, times negative binomial with a certain K-mer coverage, that's the mean, okay? And some variance that will be modeled as K-mer coverage divided by overdispersion. And then the second peak, so we will just sum them up, will be beta, which is the relative contribution of the second peak, times negative binomials. Now, genomes actually are stoichiometrically accurate. So if it's something in the genome once or twice, it will have twice as high coverage. That's how it works. So the second peak can be really modeled as twice as high coverage as the first peak. So notice that this is one parameter only, complex model, but as a single parameter. And we use the same trick for the overdispersion. So instead of keeping it constant or keeping it single parameter, which would force that every single distribution would have the same overdispersion, we will actually scale it with coverage and divide it by the overdispersion. And if you formulate it like this, it will naturally take care of that phenomenon we were showing earlier, the higher UI with coverage, wider the distribution gets. So this is just a very cheeky way how to formulate the model to keep only very few parameters while actually fitting the relatively complex thing. So in this context, because this is a probability, we expect that alpha plus beta will be equal to 1, because if you sum up a negative binomial, it always needs to be 1, because it's a probability distribution. The K-mer coverage is K-mer coverage. Overdispersion is just the parameter that scales. So this is just to emphasize. Does everybody get that if you sum up all the values of a probability distribution, needs to be 1? Yes or no? Kind of yes or really yes? It's a probability distribution. It needs to, right? It's describing certain probability space. It literally gives you the probability of all the possible outcomes. So if it's the complete space, something will have to happen. So the sum must be 1. statistics, probability. So that obviously will not work because what we're actually modeling is the frequency, not probability. We would have to normalize it if we wanted to do that. But we also are interested in genome length, so we couldn't just get rid of the absolute values. So what we will actually do is that we will model it as a frequency and just multiply it by another factor, will be the length of the genome. Okay. So it's getting complicated, but not out of hands, right? So length is a factor that will scale the probability distribution, which will, and more or less will correspond to the haploid genome size. So we ignore every repetition in the genome for now. That's fine. And they represent large portion. Yeah, okay. So what you actually don't want to do is when you do this kind of model to interpret this length as the genome size, because truly the repetitions do absolutely cover large parts of the genome, right? We actually intuitively all know that. So we cannot just take the main bit. So what we have to do instead is just to use the same trick as we use for sequencing coverage. What we do is that we take the sequencing yield and divide it by expected genome size, right? That's, again, it shouldn't be surprising to you. If you flip this equation around, just moving terms, or you will end up that the genome size is actually the sequencing yield divided by coverage. Okay, everybody with me? The same works for KMS too. So genome size is the sequencing k-mer yield, which is the total number of k-mers that correspond to the true genomic sequence, divided by k-mer coverage times quality. We already had that equation there once. Does that make sense to you? So it's very important, and you fit the model, but the estimated genome size should not really be the length you fit. The estimated genome size should be your estimate of total number of genomic k-mers, and then divided by k-mer coverage times probability. And the k-mer coverage is something you fit, and that's a lot more accurate. And to get the sequencing k-mer yield, you can actually do it in R like this. You will just simply sum all the coverages and k-mer coverages and frequencies, right? If you have 40x, or well, I don't know, you get the equation. So that's it, actually. This is it. If you will take this equation and retype it in R, it will be working. It will work. I did not really elaborate it on this alpha and beta, and this will be the last bit that I will actually change on the equation, just because it will immediately translate to heterozygosity. We intuitively understand that the more heterozygous genome is, the greater the 1n peak is. Again, can I have a head shakes? But how it actually corresponds? Well, that's actually not that difficult. Imagine two variables. The probability of a heterozygous nucleotide. Okay, nucleotide, heterozygous nucleotide, and a k-mer size. And that's a fixed parameter, right? So that's not really a variable, that's a parameter. So if we do 1 minus r, it will give you a probability of nucleotide being homozygous. If you do 1 minus r powered by k, it gives you a probability of homozygous k-mer. Okay? And if you do 1 minus that, you will get a probability of heterozygous k-mers, but because each heterozygous sites will generate you 2 heterozygous k-mers, you will actually multiply it by a factor of 2. So if you replace alpha by this term and beta by this term, first of all, you will reduce the number of parameters by 1 again, but also you will estimate R, which is your estimate of heterozygosity. I would like you to appreciate that that's some sort of magical number that appears out of nowhere, and people are always wondering what the heterozygous, like how you can actually guess the heterozygosity, right? And it's just a very simple probabilistic model. Obviously, if you start considering also duplications or polyploidy genomes, these terms will get really long and very messy. But the principle remains the same. The principle boils down to this, okay? So if you retype it in R, it looks like this. You do NLS because it's non-linearly square method. So this will already give you a hint of what the heuristic is actually optimizing. What is it optimizing? It's the residual squares. the model needs to minimize the residual squares. It's least square method. And the y will be your frequencies, and they are explained by this nasty alpha term times negative binomial, where the x will be the coverages, and the size is now these parameters that we will be actually fitting. And then we have the other negative binomial times length. So because it's non-linearly square, you need to tell it where to start the search. So you just give it a list where you just say, I will start with heterozygosity 0, with K coverage 50, with overdispersion parameter 0.5, which is actually oddly really good value. And it just turns out that whatever you do, you will not figure out anything better than 0.5. Don't change 0.5, it will collapse. And some reasonable estimate for genome size. And you just give it some sort of settings for the iterator of how it should behave. You don't have to overthink that, but you can dig in it if you would like. Like you could, it's almost like a Markov chain. You can play with it. How exactly the optimization algorithm works. It goes really beyond this tutorial, but it's like philosophically not complicated. It's just practically complicated. Right, and because I played a lot with that, I just wrote myself a tiny R package where I pulled lots of small functions to manipulate chemospectra and plot them and stuff like that, which I call Genomescope. It's like extension of Genomescope. It's not published, it's nothing, it's just a GitHub repository. But if you will be playing with chemospectra in R, it might be handy for you to look at it. There are lots of things explained, or like it makes your life a bit easier. And the code is very simple. It's very base R, so it's not like anything is hidden in C functions. You can always see what I've done exactly. So normally I would say, we actually have plenty of time. Do you want to try to fit your own models in R manually? Okay, then I will give you 16 minutes. Is that okay? And you can play. I will be answering things in the meantime, and then we can go through all the weird chemospectra we have seen at Sanger over the last few years. Actually, a lot of them are even older because I'm doing these collections for a really, really long time. Anyway.
Right, I'm just going to pause the recording there so we don't have a massive blank spot in the recording.
We have 3 people streaming on YouTube.
It peaked to about 6 earlier.
Wow.
We're now successful YouTube streamers.
Ching. Would it be like a bizarre live podcast to talk about chemo spectra and genome sequence?
Yeah, I did think about suggesting that at one point, that we do have like a bioinformaticians and coffee podcast sort of thing.
We used to do uh, this a podcast on with our improv troupe in Lausanne, where we just each of us took a paper and sort of figured what is inside of the paper. Then we gathered for 2 hours, and and we recorded what we were doing, and we just one of us explained the paper, and nobody else heard about it before, and we just improv a small scene and on like trying to reenact in a completely different settings the point of the paper. And we almost always terribly failed, but it was so hilarious. And we didn't have many people that were listening to our podcast, but like we had like 20 people that were hardcore into it.
Nice. There's a very niche market, comedic scientific paper improv group.
Yeah, it was called Science Beer Review.
I've just found it. That's my tonight's nighttime listening then.
Oh my. Also, this is during my PhD. That's almost like a decade old material. Genomescope is still running on HTTP, the web server. I'm sometimes forgetting of the luxury of living in a place where we actually have web custodians that actually do all that for you.
Did you not have that at the previous place? Was it all just you sorting it out last before?
Yeah, I've actually never seen any other place that would actually have the luxury.
Oh,
And
really?
come on, Genoscope is called, well, it's no longer called Spring Harbor. It used to be called Spring Harbor. Uh, where is Mike? I'm live streaming, not knowing of where my collaborators reside. And John Hopkins. It's a big university, like good university. I would imagine that they would be supported with that.
I think Matthew and Goyin could set you up. They've set up quite a few of our web services. So I bet they could hook you up with a good one.
Yeah, well, we have the Tree of Sex webpage that was transferred to like, just Sangha, uh, and they said like, hey, it has all these problems. We just run them, like run it to our standard QC. We fixed it for you. You're welcome. It's like now it works in mobile phones. We updated the certificates. It's all secure now. It's like okay.
Wow, okay. Last time I tried to do something like that, well, actually, I don't know, we're on stream. I don't know, I shouldn't bash the company. But no, they wouldn't, it didn't help me at all last time I tried to do it.
I probably should have mentioned that everything that I'm mentioning here as a tool or anything is actually installed within that environment. Not even things that are referred within the tutorial. So you can actually use the Git port outside that. You can just play. It's a it's a good sandbox. Okay. We went through 70 slides already. Wow, very efficient. I'm bored. Does anybody have questions? Maybe I will update the slides. Let's find more weird famous picture. I've seen some fungi that were funny.
Probably have a look through TolQC and find quite a few.
Maybe actually that's something I could add.
I suppose for those of you that don't know, we have
Well,
yeah, okay. Kimmel's on it. For a few people that have just joined us on YouTube, we're not like we are working. We're working through an exercise on the Git pod. We're not just sat here in complete silence, like complete psychopaths. We are working.
Okay, let's do that. I don't know if people noticed, but I linked also through the GitHub the Google Slides. So all those materials will be available to you. Can we add bits to the feedback form? What I would like to have there, like an option for people to upload Gamer meme. My dream is that we will have more Gamer memes.
Sure, I will.
Like the other one.
Are you just making all of these?
Yeah.
Okay.
The weird bit is that I'm not even that much into the memes. I just feel like it's like there's a bizarre joint of gamer profiling and okay, it's the time to continue, I think. Did everybody manage to get at least a tiny bit manually modeled? Did you manage to get histograms in R? Did you manage to plot it? Did you plot it yourself or did you plot it through the package? I should have had yes, no answers. But it's okay. Did you actually run the model? And did you give your values? Did they make sense? Do you feel like you don't need Genomescope anymore? You could actually feed it yourself. The truth is, in very simple cases, you actually really don't. Like it's really exactly the same thing for the specific cases. The beauty of Genomescope is that it just packages everything for you. You don't have to overthink it, and just does all this like beautiful parametric search and sliding to masking, and just gives you the model right away. But it's you should understand how it works, and I'm glad that you probably do know now how. You do know now. Okay, so. Are you ready for the fun bits? Um, okay, so in this last bit, uh, we will first go through like really, really funny spectra and just discuss what they're about. I would encourage you to unmute yourself and jump in and try to explain what we are seeing. Uh, Seriously, there is no better way how to learn than just get a few things wrong at the beginning. And I hope that you will enjoy it. So before we proceed to the freaks, I would like to tell you that the good news is that there is a home for all the freaks. All the data that are generated by the Tree of Life, which is actually tons of data, like a tons of data. We generated some form of data for 5,500 species at this point, or 4,500 species. And they're all publicly available. You can just go to this webpage that's called TalkQC, where we're just gathering QC plots for all the libraries, and you can just look like so. This in this case, we just look at I don't even know what species that is, but you can just write there what species you're searching for. You just tell you whether we have sequenced or not. You can click on that species, and it will just give you a list of libraries we have. And if you click on the library, so this is how the library overview looks like. It just gives you all the plots, so you can just simply look at them. But you can also click here and fetch the histogram and play with it yourself. I don't know, for instance, if you would like to fit the more weather or, whatever else would you like to do. And yeah, it's all thanks to heroic actions of Shane that we have this at our disposition. But it's a magnificent resource. I would definitely recommend, you know, when you want to doom scroll in the evening, doom scroll through QC. Um, and the second thing I would like to cover while we will be looking at these freaks is the principle of stoichiometry. I don't know how much I can emphasize this or how much I should. I actually think I should a lot because I think this is the bit people don't really appreciate. The k-mers that occur in a single copy in the genome will have coverage that is half of the k-mers that are present in two copies. So those are homozygous. But I would like this to be obvious and internalized. There must be absolutely no place in your mind for a doubt about that, right? Why does that work so well? Well, because you have millions of k-mers, and the probability that you will be anywhere beside the exact 1 to 2 is 0. Unless there is some sort of bias, some sort of biology, or some sort of technical problem that is involved in it. Okay? No exceptions. Absolutely no exceptions. It's not always important. Sometimes it's just a stupid GC bias on heterozygous sites and sequencing. So who cares? But it's always something real. You cannot just brush it off. Okay, so the principle of stoichiometry. How many genomes we're looking at? Well, two. It's two. We are looking at the stone coral and its cobiond. On the left, we see the cobiond. That's two, and actually both of them are diploid. So these two peaks are 1 to 2, and these two peaks are 1 to 2. They both uphold the principle of stoichiometry because they all come from the same set of cells. And they, relative to each other, obviously do not, because that, it was a mixed sample, right? If you have different tissues with different karyotypes, obviously they will have different coverages. All good? Look at this begonia. What do you think is going on in here? It's it's so what happened is that they had this begonia plant and they took a leaf, they sequenced it, and they found out that they don't have enough coverage. So they returned to the same begonia plant and took a different leaf. They sequenced it again. What I didn't realize is that in that small pot of begonias, they had two different species, and they were closely related, and but not the same. So when they sequenced them, there was this orange species with lower coverage, the purple species with higher coverage, and they were fairly closely related. So they had a bunch of chemicals that were overlapping. So that's a black distribution, and it just created this like absolutely weird thing. But when they sequenced it, people really thought that this is the heterozygous peak and this is the homozygous peak because they were not thinking about blood stoichiometry. Obviously, it can't be. How it can be 1 and 2 peak and 2 and peak if they're not 1 to 2? They are not. It was actually Luthia who figured all that out. So I actually think that's how we bonded over. Like when we have seen some weird data sets early on, we've seen some stuff. Okay, a completely different technical / biological challenge is that some of the technologies have certain biases. The GC bias of Illumina is very well known, right? What you might not know is that the PacBio also has a bias, but is quite very specifically on GA-rich repeats. Well, certainly even repeats. Is a regions within the genome that are GA-rich. Nobody knows fully why, but it just turns out that if you have too many of them, it just messes up with the sequencing deck. In most of the genomes, it makes very little difference, and so you could notice it on a big scale like a k-mesh spectrum. But in fishes, in many fishes, it's actually fairly common repetition. So it just turns out that it depends on which fish you look at, it will be more or less of a problem. So it will just create this characteristic bridge in between of error k-mesh and the genomic k-mesh. It's just simply the real genomic k-mesh, but with a dropout. It makes things more complicated. There is absolutely nothing you can do about it besides the fact that by looking at the spectra, you will actually know that it will be a problem. Okay? So this coverage bridge can be biological, as we've seen it with fishes. And I can guarantee you that because we have sequenced hundreds of fish samples and in multiple different sequencing centers with multiple different preservation techniques, multiple different library preps, and it's always the same thing. But here, I have an example of a bee. It was the same lab that did the same technology on two different specimens of the same species. And in one case, they had this okay-looking chemospectra. They fitted the right genome size of a honeybee, which is approximately 230 max. The chemo coverage was 22x, and it all looks good. If you look at the right one, surprisingly the coverage is actually higher, right? So given this coverage, you would imagine that it would be super well separated from the errors, but it's absolutely not. And what more, if you fit that model, you're missing about 100 megs of sequences. There was something that went terribly wrong in sequencing of the genome. I actually think I have a bit more slides on both the begonia and the bees later on, because when I was putting together the freak show, I was combining two different presentations. These sequencing bridges do not have to come from any technical or even biological issue of the specimen prepared, but it just simply can be because the specimen is not actually a single specimen. So in this case, when we sequence the stylops, there was one clear genomic peak over here, and then this long bridge of all the other frequencies. And it just turns out that all those minor coverage peaks correspond to site frequency spectra. So if you're a population geneticist and you would be subtracting sizes of those bars, you would actually see the frequencies of alleles. So what turns out that happened is that they sequenced what 11 individuals. You can actually even count the peaks and say how many individuals they sequenced. And obviously, you need to know a lot more about your genome and what you expect, and to be able to make this assessment. But I what I actually think happened is that they just sequenced a female that was mated, and probably multiple times. So I imagine that it was not only a single male, because then there would be just 4 genotypes. Anyway, okay. So let's talk for a bit about genome size. This is hopefully a problem of the past, but in this scary example of how things can go wrong, you will also see another reason why not to use jellyfish or KMC. So marbled crayfish is a triploid genome made in Germany, really. It's like aquarists were breeding lots of varieties of the crayfish, and they just created a parthenogenetic one, which now became invasive and invaded all the way to Madagascar and just ruining everything. So, well done, German aquarists. And well, anyway, people sequenced it because they were interested, and then they did the genome. If you do the genome profiling, it looks like this. Like if you go like naive jellyfish and a Genomescope, and you see that you're missing about like a gig of the sequences. And so we were thinking, well, what's going on? It just turns out that if you estimate the coverage, the genome size like this, you're missing out. It's a the k-mers that have very high frequencies will affect this equation the most, because they sum up to very large numbers. And it turns out that some of those k-mers have 7.3 million genomic copies. So if the k-mer coverage of that k-mer would be this number times a k-mer coverage. That's like a very large number. So, and the problem with these k-mer counters is that they by default stop counting at 10,000. So instead of counting 7 million, I counted 3,000 copies. And this is true for all the high repetitive k-mers. And if I fix that, if I actually say I would like to calculate all the k-mers up to few million copies, it actually did estimate the genome size exactly what I expected. So it's basically just capturing the real repetitiveness of the genome. We actually like played a bit with there was like a recent expansion of transposons that seems to play a role in this. And we did the same with the firebelly toad. So you've seen the chemospectra and it fitted well, right? With 8.1 gigs. if you would just stop counting at 10,000, you would end up with a genome estimate with 3 gigs. And obviously you would miss, like, that's not very helpful, is it? So how to avoid that? Well, if you're using FastK, it is using this funny trick that in the last coverage, it stores as like a dump bucket. So for KMC and jellyfish, the last bin is how many KMS have that coverage or higher. And in FastK, the last bucket means how many KMS would explain the repetitiveness of all KMS that have this coverage or higher, which means that it sort of compensates within the equation for the fact that you're not counting them explicitly, which means that the genome size estimate will be actually right. So FastK did it like that. That's the smart way to go about it. That we just have this dump bucket bin that although you don't explicitly know how many times individual KMS were occurring, you know with their total sum. You basically keep this equation for all the repetitive KMS intact. So if you use FastK, just don't worry about repetitiveness. If you use KMC and you have repetitive genomes, you will have to use parameters to just say, hey, calculate me all the very, very high numbers too. And it's a bit clumsy. I would say for very big genomes, you should use FastK anyway. It's faster. There are lots of funny problems with ploidy, you can see. This is Saccharomyces. Here, I just wanted to show that because of the equation of the genome size, if you will be increasing ploidy but lowering chimer coverage estimate, you will be on the same number. So if you fit a diploid model that converges on 34x coverage, you will have the same genome size as if you fit a tetraploid model and fit half of the coverage. And it obviously makes sense if you internalize the equation for the genome size, right? Because what was in the denominator? It was ploidy times coverage. If the term is the same, obviously and this is actually this is a Deloitte rotifer and Adenita Ricci, and I just want to show that because nobody actually has a good explanation for that. Me and Ruben Novels have spent hours staring at this plot, fitting millions of different models and discussing what it could possibly be and or mean in the context of this genome. We never came up with anything remotely sensible. So the stoichiometry must work to some extent, and you see that it basically there are things from 1n up to 8n, and they all look like very well spaced, well behaving. But it doesn't make any sense because this genome has only 12 chromosomes, so it even technically can be octoploid. And this is just weird. This is a Deloitte rotifer. It was not done on a single individual. It was done on a pool, but it's also very, very unlikely that because it's an asexual species, there was all that much diversity. And you cannot actually explain it by population diversity, because if the population had a variable number of chromosomes or variable ploidies or whatever, you would never see them evenly spaced. Do you remember when the stoichiometry works? When the stoichiometry works? If all the content is within the same cell. Otherwise, it just goes by the relative frequency of the cells. Right? I would just you to appreciate that this is weird. And maybe there will be a good explanation at some point. Yeah, I see. So you've already seen this contamination plot. And you've already seen the begonia plot. Yeah, these are the two leaves they sequence. Actually, yeah. And this is the B. So it's to add to the B example, they have this like whole paper about how this one asexual strain is weirdly different and how it's all just absolutely nonsense. Like I would be mean to the authors because I actually emailed them about it and they never even bothered responding. It's just all made up. It's just a broken library. Yeah, what is this properly? Okay, that's it. These are the freak genomes. I think we have a bit of time left, so we will either look at your game spectra or we will be discussing things. But before we finish, I would just let you know that there are more resources that we generated. It took me, well, not me, it took us about 8 years to get this by-archive paper together. It's a 17 pages of pure Kamer gold. There is everything from the history of why it's called Kamer to all those statistical properties that you now heard about. And I think actually it might be right now a good time for you to read it because I think now uh, the, that you soaked in a bit of those intuitions, it might make a lot of sense to you and it might be an easy read. We have an accompanying tutorials. I don't think I actually had anybody properly testing them. So if you go through them, actually find an error and report it, it would be absolutely amazing. And that's it. Thank you for your attention, and let's discuss what you have.
So I just posted the link to your tutorial in the chat.
Is this working?
It is, it is. We hear you. I don't see you, but we hear you.
You know, I think that's my wife on a phone call.
Excellent. Well, if you're all content, we can also just leave it in here. But hey, I'm getting about an email a week about like weird chemistry structures. Could you please explain it to me? I kind of suspect that some of you are actually one of those people that are sending me those emails. So wouldn't you mind just showing it now that I'm sitting here? Come on, I'm sure you have all weird chemo spectra to understand. Or should we respond to latest GitHub issues we have? We can close a few tickets together.
I mean,
Can we get some real life examples?
Wouldn't it be embarrassing if it actually turns out to be yours? Huh? I actually,
Hello, sorry. I was just thinking before my personal experience that I've troubled with, but I think you already answered with the presentation was work with a lot of plants and the genome size estimation when using Genomescope was usually a bit off. So that had me then resorting towards just I used the total amount of bases I calculated from my raw reads and the K-mer coverage there to do the calculation. And then I got to a very close estimate what I got from the assemblies. But now I'm thinking it's probably because I was using KMC that it was maybe with that frequencies that
yep.
disconnection came in. So I'll now try running them all again, and then they should probably work a bit better out. Or was there something different?
That's right. And also in a week's time, I will be presenting smudge plots. So if you have strange ploidy levels, it's a different diagnostic tool based on KMS. The advantage of using FastK is that it's actually smudge plots is now using internally FastK database too. So you actually don't have to redo the same read set twice. You can just make one KMA database and you can just give me the histogram, give me the smudge plot KMA pairs. And actually, Gene Myers, who wrote that KMA counter, rewrote almost all the tools you can think of to be compatible with FastK. So, for example, you can run Mercury or some of the features of cat all on the same database. So I, I guess I'm collaborating with Jeans. I'm sort of also trying to make him happy by promoting his software, but truly it is the fastest. I would say it's not uh, obviously and objectively better in all ways to KMC. I think these two are like two good contestants, but I think especially for the genome profiling, it's just safer to use FastK because it just simply gives you the estimate right.
Okay, perfect. Thank you
And
very much.
And to go a tiny bit back to how you exactly, so if what's the difference between doing it by the estimated k-mer coverage and just calculating all the k-mers in all the reads? Well, it's because when you fit the model in Genomescope, you can exclude the left side residuals, and this is what is called errors. So Genomescope doesn't have an explicit error model; it has an implicit error model. So you basically exclude that from the genome size estimate, which will make it a bit more accurate.
Okay. Thanks.
I have a question that I'm not sure is like completely related to Genomescope per se, but I was wondering if there was an easy way to kind of extract the reads where the k-mers that are in specific peaks come from. Like, say, if I have a haploid peak and a diploid peak, is there an easy way to say, okay, extract all the reads that have given haploid k-mers?
Actually, I think there is. I think I wasn't so there is this tool called Cookie Cutter that allows you to find all the reads that contain a sequence. I think extracting so you can just say you can say give me all the k-mers that have coverage between 100 and 200x, and then it will give you a k-mer dump, and then you can say give me all the reads that contain this any of these k-mers in the list. But think about the challenge of well, each read does not contain only the generic k-mers or 100 k-mers, it contains everything, right? So your question is, how can I extract reads that are in the 1N peak? Well, you probably can, but it's probably most of the reads will have both 1N and 2N peak k-mers, right?
Mm-hmm.
So this kind of separation would make more sense for, I don't know, things like the stone coral, where you could just say, give me everything that is in the rightmost peaks. So I actually get that subgenome out of the sequence.
Okay.
So in that case, you could use the things like the cookie cutter. There is also, we were just talking with at the Sangha assembly chat about how to should do this more. I think if you will stay tuned, you will hear more about that.
Okay, cool. Thank you.
Anytime.
Is this cookie cutter comp bio? This one we're on about. There seems to be multiple cookie cutter projects.
Yeah, there are, and there probably will be better ways just to explore. And I think FastK also has a way how to map k-mers on reads. So then you can actually be looking at the profiles of reads, so, which is pretty neat. Like a k-mer coverage profiles of reads.
A bit of a follow-up question for my understanding. You said that a read would have k-mers that are both in the haploid peak and in the diploid peak, but would that just come from like randomness? Or because if you have a haploid read, I would expect all the k-mers to go in the haploid.
But what does it
peak.
mean a haploid read? Like where in the diploid genome would haploid read come from?
Like if it's a male and it has an XY.
Oh, sure. Okay. So now I see where you're going. So I you can subset. So you right that if you are trying to get Y-linked sequences, then the k-mers that will be hitting Y will most likely be in 1N copy,
Mm-hmm.
unless it's repetitive, and which actually is fairly common on the Y chromosome. So again, tiny bit of a challenge. Um, unless it's a pseudo-autosomal region. I guess what I was thinking is majority of the gamers that are 1 and or 2 and are not from those heteromorphic sex chromosomes are from heterozygous sites. So in those regions, some bits that are homozygous will be shared, and so they will be in 2n copies, and those that are heterozygous will be in 1n
Right.
copy.
Right, I
So
see.
that's why I said that most of the reads will actually have both. But you're right. So uh, I spent tons of time thinking about that and actually working with that and trialing of how to subtract sequences on different levels. So we quite often did this cat comparison of multiple libraries, and then we just say, give me all the k-mers that are in 1n copy in male and in 2n copy in female, or like I did all those combinations, and then we just took those k-mer dumps and we sprinkled it on top of a genome, or we sprinkled it on top of a assembly graph. We tried all that, and the challenge was that you start mistaking the genetic variability with the sex chromosomes. That it will be a mixed signal, and for like stupid species like Pierre Rapis, where you have like more heterozygosity than the span of the sex chromosomes, it will just brutally dominate the whole bloody signal, and it just becomes a real mess. So I really like your thinking, and I would be very happy to uh, chat more about exploring how to do that right. I actually did not figure. Despite actively thinking about it and working on it, I still haven't figured out like a good universal way how to do it for
Difficult
species.
problem.
Yeah, it is. Well, but for many species, we actually did that right. I don't know if you've seen any of my papers or our papers. We did something like this for the German restricted chromosomes in the fungus nuts.
Yeah.
That was dope as hell, but it was a lot simpler problem because uh, it was a lab line. It was completely like it was an isoline. It's 100 years old. By the way, that isoline was isolated by Charles Metz, a PhD student of Thomas Morgan in a Cold Spring Harbor in a chicken coop. It's a it's 100-year-old lab line, and we sequenced
Mhm.
it. History. Um, we did the same with B chromosomes with Phenococcus viberni, where we had 0B, 1B, and 3B sequencing runs. And by looking at the sort of the coverage ratios, we actually managed to find which sequences were in what copy number on autosomes versus B chromosomes. We figured that in partially in some systems where we spent time staring at it, and when we like did the careful QC and did the right thing, but getting it automated and get it right is that's the challenging bit. But I think you might be onto something by looking at how the consecutive k-mers on a read look like. Because if you will look at individual reads, then I expect that it will have a different pattern on the autosomes and X chromosomes or sex
Mhm.
because you expect that all the consecutive k-mers should somewhat behave. But there's the repetitive. I don't think it has a good answer. You're triggering me.
No, thanks a lot. Yeah, it explains things.
Well, now I need to Google who you actually are. Uh, hey, you're with Laura. Lol. Sorry, I didn't connect. Of course, I said like I know that name, and I do know that name. My
Yeah, no,
apologies.
I was coming at it definitely from the fungus gnat perspective.
Now, in fungus is actually easy. It's uh, I would even say that you don't truly like in fungus nuts. I don't think you need to extract the reeds. Well, unless wait, this should not be streamed on YouTube. It depends which species you're after. If you're doing some of those midges, I think it might be um, more of a challenge.
Yeah, I
It's
think,
in series is definitely easy.
yeah, I'm working mostly on Cyrids.
Ah, it's good. It's good. But do you have a species we don't? You know that we have now a
Um,
bunch of different chromosomes.
well, I'm trying to basically figure out, the X inversion in Bradisia cropophila. I'm trying to figure that
Yeah.
out for a different species that is also supposedly has this inversion.
Which one?
Impatiens?
I don't think impatiens has that inversion. Do you have our data?
Yeah, we did the same. I can send this to you actually. So we did the same kind of like heterozygosity plot on our impatiens line from like collected in the UK, and they do have like a huge like plateau of heterozygosity from
Oh, okay.
a female producer, yeah. But
Well, you can see that.
I know that Rob has like a very different result, I think.
Oh, so well, impatiens is really wacky. So if you actually look at the old papers of about impatiens, they report lots of different things. I think
Mm.
it's one of those lines that is not very stable. And if you look at the germinal chromosomes of impatiens, they also look very, very different to all the other species. They're less synthetic. They have a lot more turnover of material from the core genome. It's a whole different story. I think impatiens is a it's a it's a wacky genome. That's
Yeah, that's why I'm interested in trying to like look more into it.
yeah, fair enough. Good. More came questions. I think we lost a few people in the audience, Melanie.
Sorry for ruining your talk.
I think Damon will need to do some editing.
Uh,
It's not
sorry to bother again. I was just wondering, do you have maybe a good suggestion? Because you mentioned for PacBio to use K31, but currently I'm working on ONT, which still has a bit more errors within it. So I've dropped it down to 21, or what would you say there for the, or
Then
bump it up to
just
31 again?
spend your mental energy on things that matter. Just that's a it just you understand the principle of coverage. You understand the principle of errors propagated in KMS. Just do one, look at it. Does it look okay? Just
Okay, so it's not like that it would skew the data in a sense, because
no.
I was
You
now
should
just wondering with that.
you should regret every single moment you spent thinking about it. It's not the time well spent. Yeah.
Okay, thanks.
Drive.
Right, in that case, do we have any more questions and we can get back 4 minutes?
And then if you
No,
click on the
cool.
triangle,
Right.
you don't-
Well,
Wow.
thank you all very much for coming. Thank you, Camille, for giving up your time to give the session. And I hope to see you all in the next one.
Wait, one question. Are you coming in a week?
But I need to put the new. I'll put the.
Am I coming in the week?
I need to put
What?
In a week, there's Smudgeplots release. It will be epic,
Oh,
like absolutely
yes.
epic.
Yes, everyone should absolutely come to that.
Hey, good.
on
See you in a
YouTube.
week then. Bye.